All right, welcome to our honors colloquium here on April 14th. Very excited to be hosting one of our graduating seniors in the honors program, Ms. Anil Pournouri, a biology student, who's going to be speaking on hypertensive stimuli increase, lymphangiogenesis in human lymphatic endothelial cells. So, take it away.
Let me just introduce myself. You guys know who I am.
Make sure you speak up so the people on Zoom can. Oh, and let in the Zoom people who are going to stand here. All right.
So that was my student right there.
All right.
Okay, so hi, everyone. I'm sure all of you know me. If you don't know me, my name is Anil, and I will be graduating a year early, and so that's pretty exciting. And so last summer, I was accepted at the summer research program down at Texas A&M College of Medicine. So what they're doing is they basically received a 5-year grant from the National Institution of Digestive and Diabetic and Kidney Diseases. So you're choosing 10 students from all of the A&M schools across Texas. And so we had about, I think, 8 or 9 students. They do have like a general application that's open to the rest of the universities across the country. So we did have students from, California, Georgia, Oklahoma. Somebody was from Canada. I don't know, but it's a very good program. How many times are you?
Can you check the chat as well? All right, that's from Caitlin.
Yeah,
Thank you.
she's taking attendance first.
All right, go ahead.
So yeah, it was a 10-week program where we got matched with one of our top 3 faculty members. So what they did is they gave us a list of professors and we had to choose one of them. And so for us drummer students, as we're minority and underrepresented students, we were limited to, research that was, basically from like digestive or diabetes. And mine was about hypertension. I love the cardiovascular system. If you're one of my students, you would know. And so, before I get into this, if anything does not make sense, please do not worry. It took me the whole summer to connect all of the dots. And if you have any questions, please ask me. So this research was going on 2 years before I joined, and the PI of the lab, which is just the person in charge, said that we joined at the last chapter, so we helped kind of close it. And so let's get started.
You'll have to click on the screen, yeah.
Okay.
You can also close the little box with our picture on it. That's fine. There you go.
All right, so as you guys know, hypertension is high blood pressure and it's the most common cause of cardiovascular deaths in the United States. Now, lowering the blood pressure by just 10 millimeters mercury can reduce the risk of death and improve patient health outcomes. Now, we have so many improved treatments, we have so much awareness going on. Still, 30 to 60% of the patients cannot reach their target blood pressure. So that is a big problem, and our lab is hoping to find a treatment that can either prevent it or fully treat hypertension. Now, previous research by our lab showed that the renal infiltration of activated immune cells actually contributes to hypertension and inflammation. So your kidneys do such an important job. So they filter out plasma from your blood. They also reabsorb useful materials like water and solutes, and they secrete the things that you don't need in your body, like metabolic waste. And if there's content that is left or there's too much remaining, that is going to be excreted through the urine. Now, the diffusion of activated immune cells through kidneys can actually contribute to hypertension and inflammation. So that is what our lab found out before I joined it. And that promotes sodium retention, which is basically like if you eat too much salt, your body is going to retain even more salt and it's going to increase the fluid that is outside of the cells. Now, when that fluid increases, your body once again is going to retain even more salt and fluid. And sodium retention itself also contributes to hypertension. And it can promote renal injury. If you see any word that has renal in front of it, it just means kidneys. And renal fibrosis, which is the consequence of almost all of the progressive kidney diseases. And so another research, which is not by our lab or anybody at the Texas A&M College of Medicine, it showed that mice that lack certain immune cells, which are macrophages and T lymphocytes, those are very important white blood cells in the body. Macrophages, they engulf the foreign substances in your body and they initiate an immune response. whereas T lymphocytes will directly kill the host infected cells. So these are very important, especially when it comes to, any viral infection or anything in the body. They basically kill them. So now mice that lack those certain types of cells are more resistant to blood pressure elevations, which this suggests a key contribution of these immune cells to hypertension. Now, I did not know I would like the lymphatic system at all until I started to work in this lab. So you have vessels that drain the extravasated fluid. That is the fluid outside of the vessels. Anytime there's a fluid surrounding cells, capillaries, or vessels, that's called extravasated or interstitial. So they drain that along with the proteins and the immune cells in that fluid, and they transport them to the lymph nodes. So let's say there's inflammation in the body. We have two different types of inflammation. We have acute inflammation and then chronic. Acute is when your body responds to like a sudden damage. Let's say you cut your finger accidentally. So the body is gonna send those inflammatory cells and they're gonna initiate a healing process. Now, chronic inflammation is when the body keeps on sending those cells, but there's no outside danger whatsoever. So in acute inflammation, your body, as I mentioned, is gonna respond to it by dilating those lymphatic capillaries. However, if inflammation keeps on occurring, it's going to result into the formation of new lymphatic vessels, and that's what we call lymphangiogenesis. That's that big word in my title. It's kind of scary. And so, yeah, that is very important. So we used 3 different hypertensive stimuli. We used angiotensin 2, which angiotensin itself is a peptide hormone that constricts your vessels. And angiotensin 2 in your body is used to, elevate blood pressure. It kind of maintains that and also maintains like homeostasis along with fluid balance through different actions. And then we use salt. That is not the salt you put on your food. It's another salt. They use that very commonly in labs. And then we also use asymmetric dimethylarginine, which is ADMA. That is a naturally occurring chemical in your body that is in your plasma and is produced through like different metabolic reactions. And as I mentioned, these cause the constriction of vessels and they also promote sodium retention. And as I mentioned before, these changes are actually accompanied by the inflammation of your kidneys, along with the activation of immune cells. Now, with other states of inflammation, the lab saw an increase in the renal lymphatic vessel density in the hypertensive mice. So those vessels, the density of them started to increase. Now we know that we can prevent and treat hypertension by just genetically increasing the renal lymphatic vessels, which is going to decrease the immune cells in the kidney, and it's not really going to promote hypertension. So now that the lab knows that, we got the answer to one of our questions, they're starting to wonder whether the hypertensive stimuli is directly causing the formation of these new vessels or not. So we hypothesized that the hypertensive stimuli will directly increase lymphangiogenesis. So to see if the hypertensive stimuli induced any cell proliferation, which is just an increase in the number of cells, we started to grow human lymphatic endothelial cells in endothelial growth media MV2, and that is a specific one just for the human lymphatic endothelial cells. The girl I was working with, which she was also part of the research program, she's not my best friend, she was working on the mouse lymphatic endothelial cells, and she had to use another media. So they each have their own different medias, because that plays a part on how fast they grow and how confluent they are. So once they reach 75% confluency, that is very important. I'm not sure if you guys can see. Okay. So here is, we will call this 30% confluent. That's when, there's some cells forming, but it's not as much as we want them to be. So whenever we're passaging them, whenever we grow cells, it's called cell culture. And so we start by growing them in a flask, and the media is actually pink, which is really pretty. And so these little cells will give you so much trouble. Like one day you will go in and the CO2 tank that we're incubating them with would be empty. And so the cells would, it would be over the weekend and you would be like, oh my gosh, are they dead? So we can have, a mental breakdown for a minute, but then you would have to passage them each time when they are confluent enough. So it's very important on what time you passage them, and sometimes you couldn't see under the microscope at all. So if you passage them when they're, not really like 30% confluent, then you would not get enough cells in your microbubble plate, because you put this neutralizer on it called trypsin, which is gonna, make these vessels and then make these cells come off the bottom. They're stuck at the bottom. And so whenever you would passage them, they're not gonna be spread apart. And then when we have ones that are too confluent, whenever you're passaging them, they would die in the process and you would have to start all over again. And this would take about 2 to 3 weeks just to grow these cells. Now, once the cells were ready, we treated them with the hypertensive stimuli for 24 hours, and then we verified them for lymphatic specific markers like LIVE1, PROX1, and photoplanin. Now, these markers are basically just used to detect lymphangiogenesis. There are so many different specific markers that they use for different reactions. Those were the ones that we had to use because, we were just looking at the lymphatic vessels. And then we isolated the RNA, and we used this machine called a nanodrop, which you literally put a drop of the liquid on there, and it determines the quality and the quantity of your RNA. And then we perform PCR, which I'm going to get into what that is, and we use ubiquitin as the gene for analysis. So I'm sure you guys have heard PCR a lot. That's what they use to identify COVID in your specimen. We have different ones. We use the real-time PCR. And so just the steps for the standard PCR, it starts with denaturation. So your DNA is double-stranded, and during this process, they heat it up so those strands become separate. And then you have annealing, which is when they add a primer. They have specific primers. They don't use the same primer for all of them. So whenever that primer is added, it's going to bind to the edge of your template strand. And so the next step would be elongation, or sometimes they call it extension, is when they add DNA polymerase, which is just an enzyme, and it's going to add on to that primer, and it's going to keep on going on the template strand. And this process is actually continued for 25 to 35 times until you have the exact copy that you need. And for us, we use the real-time PCR, and that is, it's way faster, it's way easier, and you can also detect it while you're amplifying your DNA. Now, here is our results. You might be like, Anil, where are the numbers? I'll get into that. So we did not observe any cell proliferation because there was no increase in the lymphatic vessel marker. So that just means that the hypertensive stimuli did not directly act on the lymphatic cells. That is the opposite of what we hypothesized. So we started to think a little bit further because this is a 2D cell culture environment, and lymphangiogenesis is such a complex process. So it's not the best way to measure it. And of course, we had to go 3D. So the technique we use is not common at all. We actually got it from the lab that was next to us. They worked on blood cells. So we had to adjust so many of the protocols on there. And so it's called 3D gel invasion assay. Once again, we started to grow those human lymphatic endothelial cells, but this time we did them in the 96 well plates. And it's this tiny, there's like 96 holes, and you had to grow these cells in there. And then for the gel, we had to use various growth media. So we use vascular growth factor C and A, basic fibroblast growth factor, and hepatocyte growth factor. Now, the reason why we chose this is because they actually induce lymphangiogenesis. And for the basal media, we used fibronectin, which just aids in cell proliferation. So whenever these cells were ready, we had to stain them with toluene blue. That is why most of my presentation is blue. And so what it does, it gives it a blue color. And so when the gels were fixed in those 96 well plates, we had to take them out carefully and cut them into 4 to 5 pieces. And that is very hard. Let me just show you guys. That is so hard. And so whenever we were going to analyze them under the microscope, we had to put them on their side so you could see both the monolayer, which is just this top dark layer, and then the gel at the same time. And so these pictures were taken through the 4x and then the 20x magnification. 4x is when you want to see all of the gel at the same time, and then 20x is when you want to analyze the vessels closely. And we also did take 10x magnification images, but that is mostly for publications. So as I mentioned, the hypertensive stimuli did not induce any cell proliferation in our 2D cell culture environment. However, in our 3D gel, it actually significantly increased the invasion distance, but it resulted in longer and thinner cells. So let me explain that. So under our normal conditions, which is saline, you see saline in a research, that just means that is your control. So as you see here, we barely see any cells, any vessels. Those would be this. Whereas with our hypertensive treated vessels, there's way more up here. And you might see some blurry ones down here. That's because whenever you're looking at it through the microscope, you're going through different magnifications. So what shows up here is not going to show up down here, but we did count those when we were analyzing our data. And so when we look at them closely under the normal condition, the vessels are actually shorter, but they're bigger. And that might look like it's bigger and longer, but that's 3 different vessels that got together. We don't know why that happened. But when we were analyzing our data, we had to count those as 3 different ones. And so here is our vessels that were treated with the hypertensive stimuli. If you see, they're actually way longer and they're more thin, which just means that there's less cells that are getting together to form them. Now, as I'm going through this, I'm going to go back to the pictures and show you. So the lumen-forming structures is basically just another name for vessels. We call them vessels, sprouts, and lumen-forming structures. And so when we counted those, we saw that it significantly increased in our treatment groups when we compared it to the control. So as we see here, there's way more vessels, and then once again, you can't see them down here, but there's like shadows of them. And for the invasion distance, that is just the minimum distance from the monolayer to the tip of the gel. That also significantly increased in our treatment group. So it means that these vessels are invading further down the gel. We don't know why. And so that is for that. And the way we measured this, there was a software called ImageJ, which like you have the picture and you would put a line on it and it would just measure it for you. And the lumen diameter, it's just this clear space here within a membrane, within a continuous membrane free of nuclei. So we had vessels that were, you know, they had a clear membrane around it and they had like different dots in the middle. We never counted those because those were just not, you know, that's not how a vessel is supposed to look. And so we took the maximum width of that using that same program, and we saw that it significantly decreased in our treatment groups. So the vessels are very thin, and which kind of makes sense when you think about it, because the hypertensive stimuli that we use, they constrict vessels. So that is the reason why we think that these are actually resulting in thinner and longer vessels. And then the cells per invading structure is basically just the number of cells that got together to form these vessels. And we put DAPI, which just stains the nuclei. And when you look at it under the microscope, you see glowing dots. And so we were in a dark room because that was the protocol for the microscope. And so it was me and my partner, we were counting the dots, and at one point you would see extra dots. So that's when we know it was time to take a break. And so we did have the PhD student to go back and double check because, you know, you would start seeing that was not there. So that was very good. And so we saw that it significantly decreased in our treatment groups when we compared it to our saline control here. So that just means that it kind of makes sense because the vessels are thinner, which just means that there's less cells that are coming together to form them. So in conclusion, the hypertensive stimuli did not cause any cell proliferation in our 2D cell culture environment. However, it caused an increase in lymphangiogenesis, and we can see that through the 3D gel invasion assay. So these vessels are invading further down the structure while they're being longer and thinner. And I would like to acknowledge everyone in the lab. Brooke was the PhD student we were working with, and she left in the middle of the research program because she got into her dream job. So it was just me and my partner. And Bethany was the lab manager, and Shobana was a fellow student who was also helping us when Brooke left. And Karina was my partner, and Dr. Brett Mitchell was the PI of the lab, and he's also the program director. And this work is supported by the NIH. And it is getting published. The abstract right now is on the American Heart Association Journal. So if you look up my name, it shows up. And so we are waiting for the paper to get out, but when it gets out, I'm sure y'all are going to see it on Facebook. And I also did present this at the A&M Pathway Symposium down in College Station, which was about, I think, 200 and more students. And I placed second in the biomedical and health sciences undergraduate poster competition. I also presented it at the LSU student forum, I think. Yeah. And yeah, these were the people I worked with. Amazing, such a great opportunity. I'm so grateful. And thank you.
sharing. We probably have time for a question or so, if anyone wants to ask. I hear you did great. I just have one very quick one, and that is for someone who knows obviously a lot about lymphangiogenesis, clearly as a medieval historian, but for everybody else, what would be the application from a medical standpoint of this research?
So we think because our 2D cell culture environment didn't work the way we wanted to, but for the 3D part, when we use the VEGF-C, the vascular growth factor, we think that is going to be an important role in like treatment in the future. So we're going to redo the first part. So we're going to treat the cells with VEGF-C in hopes of seeing cell proliferation. Use VEGF-C in treatments.
Okay, there we go. Very cool. Any other questions or anything before we let Anil go? She says no, you're done. Okay, well, thank you once again, folks. Thank you, Anil. It was very, very good. Very interesting.